Good afternoon, everybody. Today we'll talk about laboratory diagnosis of pneumonia in the molecular age. You know that the respiratory tract infection is the commonest site for infection. And a number of viruses seem to affect the respiratory tract throughout the year. You'll find the mention of rhinovirus, coronavirus, enterovirus, adenovirus, respiratory syncytial virus, parainfluenza virus, influenza virus, and even streptococci. Normally, it's been seen that respiratory tract infections can be classified on the basis of onset of infection, which could be acute or chronic, site involved, upper respiratory tract and lower respiratory tract, place where the infection is acquired, community-acquired pneumonia or hospital-acquired pneumonia, age of infection, childhood infection or adult infection, symptoms which could be typical or atypical, or etiological agent which could be bacterial, viral, or fungal. It's been conceded that respiratory tract infection is the commonest cause for antibiotics to be given. And they feel that most of the time, antibiotics are inappropriately used. Now, pneumonia is a specific condition where the lungs alveoli are inflamed and filled with pus, making it difficult to breathe. And that is the function of alveoli. So there is difficulty in breathing and there is cough as well as fever. As I was mentioning, that antibiotics are prescribed for respiratory tract infection, but it is felt that most of the time antibiotics are wrongly prescribed for respiratory tract infection. Nevertheless, even with the advances that have occurred in diagnosis and treatment, respiratory tract infection is the 5th leading cause of deaths. And worldwide, about 5 million cases occur, but otherwise in India, there's still lower respiratory tract infection account for 5.1 lakhs of deaths. There are a number of pathogens involved. Viruses seem to play a very important role in the childhood. But as we've seen, coronavirus SARS-CoV-2 is also a virus which has caused havoc in the world. But commonly, it's Streptococcus pneumonia, which is associated with pneumonia. But fungi, and as you would have noticed in the recent past, there was talk of mucormycosis, as well as there was talk of aspergillus causing infection. Acute lobar pneumonia is the classical pneumonia, where a person has purulent sputum, rales can be heard over the lung involved, and the patient is very sick. The common cause of acute lobar pneumonia is normally Streptococcus pneumoniae. But after influenza, Staph aureus may also cause it. In persons who have chronic alcoholism, Klebsiella pneumoniae could be a causative agent. Persons with bronchiectasis or cystic fibrosis may have Pseudomonas aeruginosa. So, though acute lobar pneumonia is normally caused by Streptococcus pneumoniae, but other pathogens may also cause the infection. If you look back in the history of what has been happening, after the typical pneumonia, there was also atypical pneumonia. And every 10 years, every decade seems to bring out a new causative agent. In 1940s, there was Eton agent, which turned out to be Mycoplasma pneumoniae. in 1970s was the Legionnaire's disease, and the causative agent was found to be Legionella pneumophila. In 1980s, there was the Tawar agent, which turned out to be Chlamydia pneumoniae. Then in 1990s, AIDS associated infection was Pneumocystis carinii, which turned out to be now a fungus known as Pneumocystis jirovecii. Then in 2000, you had severe acute respiratory syndrome. Then there was Middle Eastern respiratory syndrome. And now, of course, in 2020, and since then, we've been having SARS-CoV-2. SARS-CoV-2, it's also been reported that there are mixed infection in about 20% of cases. If you look at published literature from lower respiratory tract infection and community-acquired pneumonia, the etiological agent is normally found in, with the best attempts, in not more than 40% of cases. And majority of them, you'll find, if you're talking about lower respiratory tract infections, then viral is about 38% and 11% for bacterial and 10% having co-infection. On the other hand, if we are talking about a community-acquired pneumonia, then though etiological agent can be identified again in only 40%, bacteria plays a bigger role, but viruses are also present. So the traditional workup, when a person suspects community-acquired pneumonia or lower respiratory tract infection, is to request for sputum culture. Some persons also look for urine antigen testing for Streptococcus pneumoniae or Legionella pneumophila. And it's also been suggested that though the positivity rate is low, blood culture should also be sought. Using these methods, it's very unlikely that you would find the pathogen in majority of cases. And that's why you would find many physicians do not order any test, and looking at the X-ray, decide what antibiotic to give. If Streptococcus pneumoniae is the causative agent, then looking at the gram smear alone might show you many lanceolate type diplococci, as you see on the left panel. If you have antisera and you put a drop of antisera to that, you would find that there's a swelling reaction. And if the bacteria can be grown in culture, then you can investigate whether it is penicillin sensitive or penicillin resistant. But as I said, by using traditional method, you will detect only about 15 to 20% of cases. There are a number of viruses, as I indicated, throughout the year that have been implicated as causing infection. Influenza A, B, and C. A, B have very strong association with the respiratory tract infection. Rhinoviruses are normally associated with common cold. Parainfluenza virus 1 to 4, adenovirus, and enteroviruses have also been associated. And there were already 3 coronaviruses associated with common cold before we had SARS in 2000, then MERS in 2010, and now SARS-CoV-2 in 2020. Besides these, there are 5 bacterial pathogens which are difficult to culture: Mycoplasma pneumoniae, Chlamydia pneumoniae, Legionella pneumophila, Bordetella pertussis, and Bordetella parapertussis. They are very difficult to culture, and a routine microbiology lab would not be able to detect this. So neither can they detect the viruses, nor can they detect the 5 real pathogens. All they can find out is mostly strep pneumonia. That's why Genes, a company in South Korea, introduced a novel primer sequence, which consists of 2 portions of primer. The 5' portion was twice the size of the 3' portion, and it is linked with the linker. Since the length of the 5' portion is longer, it would have a higher TM and it will bind to specific site. Along with that, once it is bound, it will give more, so it will give more sensitivity since it's a longer portion of the primer. And with the linker, it will then bind to specific region so that the smaller portion will give the specificity. And by this DPO technology, dual priming oligonucleotide, they would get very high sensitivity and very good specificity. See the video to understand how it works.
This animation introduces Gene's innovative DPO primer technology, which has become a model for multiplex PCR in the field of molecular diagnostics. Primers are the key for successful multiplex PCR. Unlike a conventional primer, a DPO is a new and novel primer concept that fundamentally blocks non-specific priming and consequently delivers a reliable multiplex PCR. It has a unique structure that contains a polydeoxylinosine linker that is inserted into the primer sequence. This separates a single primer into 2 functional regions that have characteristic annealing temperatures. Because the sequence of the 5' portion is approximately 2 times longer than the 3' portion, the 5' portion has a higher TM and will preferentially bind to the template DNA first during PCR. The 5' portion works as a stabilizer, initiating stable binding of the DPO primer. In contrast, the 3' portion under normal PCR cycling conditions can't bind to the template DNA by itself. The 3' portion works as a determiner of specificity. Its binding is required for elongation of the DPO primer. Only target-specific extension will result from the successful annealing of both the 5' portion and 3' portion of
So DPO gets the specificity to the reaction, both the sensitivity and the specificity. The next portion of chemistry is the using of TOS. TOS is based on tagged oligonucleotide cleavage and elongation. Here, besides the DPO primer, you have a pitcher, and pitcher has a tagged portion which is cleaved as the DPO primer will increase the length, will synthesize new nucleotide. This will then release the tagged portion from the pitcher, which will be then caught by the catcher, which has homologous sequence. By changing the length of the catcher, you can differ the TM. So different different sequences could have different catchers, and then you can identify different pathogens. Watch the video to understand this better.
TOAST technology is the new paradigm for high multiplex real-time PCR. TOAST technology allows the multiple detection of up to 20 targets from a single sample in a single tube on any existing 4-channel real-time PCR instrument. The key components of TOAST technology are DPO primer pairs, pitchers, and catchers. The DPO is Seegene's proprietary target-specific primer and provides highly specific amplification of the target region. The pitcher is a tagging oligonucleotide which hybridizes specifically to the target region. The catcher is a fluorescently labeled artificial template. The DPO primer and the pitcher hybridize specifically to the target sequence. The tagging portion of the pitcher is designed not to hybridize to the target sequence. During the DPO primer extension, the pitcher is cleaved by the exonuclease activity of DNA polymerase, and then the tagging portion is released. The released tagging portion hybridizes to the catcher, which has a complementary sequence of the tagging portion. Extension of the tagging portion separates the fluorescent reporter molecule from the quencher molecule, resulting in signal generation. The signal can be measured in real-time base and/or analyzed by melting temperature base to detect the presence of the target sequence. Since the catcher is designed to have a designated TM, when the target sequence exists in the sample, the expected unique TM value is obtained during the melting temperature analysis. The TM value can be controlled by adjusting the sequence and length of the catcher. This unique approach allows for a constant TM value that is not affected by sequence variations of the target sequence. Since the TM value of the catcher is controlled by the design of the assay, multiple targets can be detected very easily in a single channel. TOAST technology allows for detection of 5 targets per channel, which results in detection of up to 20 targets on an existing 4-channel real-time instrument.
So the benefits are flexibility and agility, multiplexing capabilities, and analytical performance. There's the advantages that new tests can be developed, and the biggest advantage is one sample, one tube, but multiple results. Up to 20 viruses or genes can be identified in one run. So DPO and TOAST together will give you the benefit. Of course, any microbiology work starts with proper sample collection, and these are special sample collection tubes which have bristles, and inside this the fluid, the respiratory fluid in the soft nylon fiber, it will be trapped, and then this is transferred onto a liquid which is ENAT liquid. So the liquid will be the transport medium so that the virus can survive, is preserved in that. It is transferred between 5 degrees Celsius and 25 degrees Celsius. And in the lab, so the steps will be that first you peel off the swab and you collect the nasopharyngeal swab. That swab is then transferred onto the viral transport medium. That viral transport medium is then transported between 5 and 25 degrees Celsius. When it reaches the lab, then we use an automated platform to set up the multiplex PCR. In this, the first step would be that we centrifuge the specimen, then we load up onto the automated analyzer where this would then do the extraction of the RNA or DNA, and after that we'll set up the reaction. If because RNA viruses also need to be tested, so first step will be reverse transcriptase so that cDNA will be made, and then subsequently you would set up the PCR reaction. And once the reaction has been set up, then we'll read the result. And the first step that we have to do is to look at the internal control. Every reaction has an internal control, and normally every run must have a positive control and a negative control. So the internal control must be positive, and the positive control must be positive for all the channels, and the negative control must be negative. Once this has been met, you then look for other channels, and you find here that internal control is positive, and along with that there's human rhinovirus and adenovirus, and also parainfluenza virus 4. And so with one run you would be able to report on 16 different viruses. In a separate run, the 5 bacteria which cause pneumonia, normally atypical pneumonia, but are very important, like Mycoplasma pneumoniae, Chlamydia pneumoniae, Legionella pneumophila, Bordetella pertussis, and Bordetella parapertussis can be tested and reported. Here again, as you notice, that first you look at internal control, which is positive, and then you find that Bordetella parapertussis is positive here. So in this, you can say that we are reporting on 5 different bacteria simultaneously. One of them is positive, but the other 4 are negative. Normally, when you buy a computer, they say if you have waited long enough, you might get a better version. So we had used cGenes from 2011 when I was in the Middle East. Subsequently, I came across BioFire's film array, which has the advantage that it will look for 27 bacteria, 7 genetic markers, and viruses, etc., simultaneously after 2 minutes of work. So all it requires that it look at 34 different targets rather than the 20 that we were doing, and your hands-on time is 2 minutes, and it'll give you results after 1 hour. This is the bacteria, and it will give results semi-quantitatively, because we know that many of these bacteria are also present in the upper respiratory tract. So the sputum, when we collect, they could have some of the commensals also being collected. So a quantitative culture, a semi-quantitative culture will give you an idea about what is the load of the bacteria in the sample. Besides looking at the bacteria which cause hospital-acquired infection, it also has panel which takes, in the same panel, there's atypical bacteria. There are also resistant markers and influenza. Influenza, adenovirus, coronavirus, parainfluenza virus, respiratory syncytial virus. The heart of this is a pouch. Pouch has all dried samples, and along with all dried material are available, and there are 2 rounds of PCR. The first round of PCR So, and then after that, there'll be washing in between, and then the samples will be transferred into the second round of PCR, where it can detect the 34 different genes with amplification. Here, the detection is by using a dye which will intercalate between double-stranded DNA. Again, as I said, it's available as a pouch. So you hydrate the pouch and then you put the sample. So once the sample has been, so this is about 2 minutes of work, and then you set up the test, and the rest of the work is done by the machine. After 1 hour, you have the results with you, and the results, as I said, would also show semi-quantitative nature of the isolate. This is a much more sensitive technique when compared to bacteriology, and you would be able to pick up more pathogens by using the biofile.
The film array is user-friendly multiplex PCR. This innovative system simplifies molecular testing through a completely automated protocol, beginning with integrated sample preparation and ending with automated analysis of results. Starting a film array run is easy. All of the reagents required to test a sample are preloaded and freeze-dried into the reagent storage compartments of the film array pouch. To begin a film array run, the pouch is first placed into the loading block. Then, hydration solution is injected into the pouch through the blue inlet port on the right. The vacuum in the film array pouch automatically draws the correct volume, eliminating the need for precise measuring and pipetting. Next, sample is added to the film array sample buffer. This solution is then mixed and then injected into the film array pouch through the red inlet port located on the left side of the pouch. Again, the vacuum in the pouch automatically draws the required volume. The film array instrument is now ready to set up. The film array pouch is loaded into the film array instrument, and a barcode reader is used to enter the pouch ID. The sample ID can also be scanned with the barcode reader or entered manually. Last, the user ID and password are entered, and then the run is started. You are free to walk away since all remaining steps are completely automated. The film array displays the remaining time. Now let's take a look at what happens inside the pouch. First, the sample is moved into the lysis chamber, where the film array physically lyses any cells and viruses through a process called bead beading. Ceramic beads are agitated at high speed to break open cells and viruses and release the nucleic acids. These nucleic acids are then bound by magnetic beads, which are then moved from the lysis chamber to the purification chamber. Here, a wash buffer removes any remaining cellular and viral debris. The film array activates a magnet outside the pouch, which holds the magnetic beads in place while the debris is washed away. Next, an elution buffer releases the purified nucleic acids from the magnetic beads. The magnetic beads are again magnetically secured while the nucleic acids are moved to the first stage PCR chamber. A reverse transcription step is performed to convert any target RNA into DNA. This is followed by a high-order multiplex PCR involving dozens of primer pairs. During this first stage PCR, many reactions simultaneously occur in this single large volume reaction. The products from the first stage PCR are then diluted to limit any remaining first stage PCR primers. Next, the diluted first stage PCR products are combined with a fresh master mix and then aliquoted to each well in the array. Each well of this array is pre-spotted with one pair of second stage PCR primers. These second stage PCR primers are designed to amplify sequences contained within the products from the first stage PCR. This eliminates non-specific products associated with traditional multiplex PCR. Because the second stage primers specifically amplify only target DNA. A fluorescent double-stranded DNA binding dye is used to monitor each reaction. The second stage primers in each well are designed to detect one specific target. Organisms are identified based on which wells in the array are positive. The film array performs a melt to confirm the presence or absence of specific temperature signatures of the second stage PCR products. The film array software processes the data and makes a positive or negative call for each organism. These results are provided in one easy-to-read report. From integrated sample preparation to automated result analysis, the film array is user-friendly multi.
So by using the most modern methods, again, a number of pathogens have been identified, and still you'll find that no pathogen is detected in 62% of cases. But one thing that we want to know is that, is it a bacterial infection or is it a viral infection? And the reason is because antibiotics are prescribed, and in many of the conditions, there's no necessity of prescription. In fact, many places they have started antibiotics stewardship program, and they feel that 50% of antibiotics are inappropriate. And their aim is that we should have no antibiotics prescribed for bronchitis, bronchiolitis, middle ear infection, influenza, viral pneumonia, viral upper respiratory tract infection. These are all classified as unnecessary antibiotics, unless there is evidence of bacterial infection. It's also been seen that overuse of antibiotic is associated with emergence of multi-drug resistant bacteria. And it's also been seen that there's economic benefit of using antibiotics wisely, and you would save millions of dollars or millions of rupees if the antibiotics, instead of being given indiscriminately, are given for the target where they are required. So one of the methods that has been suggested to differentiate between viral infection and bacterial infection is measuring procalcitonin. Procalcitonin is a 116 amino acid precursor of calcitonin. Calcitonin is produced by thyroid C cells and neuroendocrine cells of the lung as a precursor to calcitonin. Procalcitonin has effect on calcium homeostasis, but procalcitonin does not have any influence. Inside the thyroid C cells, the procalcitonin is cleaved and only calcitonin is released. But it has been seen that procalcitonin is produced by a large number of cells, not only the thyroid cells, and this can be produced as a consequence of exposure to lipopolysaccharide of the bacterial cell wall, IL-1 beta, and TNF-alpha. And it's been seen that production of procalcitonin is selectively decreased by interferon gamma, which results as a consequence of viral infection. So in bacterial infection, procalcitonin is produced by nearly all cells of the body, while in viral infection, interferon gamma will decrease the production of procalcitonin. The procalcitonin starts rising after 3 hours of infection, and by 12 hours it has reached the peak, and it starts declining within 24 hours if correct antibiotics are administered. So if the procalcitonin is rising and you give the correct antibiotic, it will start falling after 1 day's gap. But if it continues to stay up, that would mean that the antibiotic is inappropriate. So procalcitonin can help you decide whether antibiotics need to be given, and if you have given the antibiotic, whether this antibiotic is working or not working. Because if the procalcitonin remains high while you are giving the antibiotics, that would mean that the antibiotic you have chosen is inappropriate. When you compare this with CRP, procalcitonin will rise only in case of bacterial infection. And as I indicated, that if it is a viral infection, because interferon gamma will be produced, interferon gamma will prevent the production of procalcitonin. So if you measure high levels of procalcitonin, it would mean that there is bacterial infection. CRP, as you see here, will rise after 6 hours and will remain higher for longer period. Using procalcitonin and different, but the procalcitonin that you measure must be quantitative, because if the levels are less than 0.5, 0.25, they said that no antibiotics need to be given in non-critical conditions. In critical conditions, if it is more than 0.5 microgram per ml, then antibiotics must be given. And in case it is less than 0.5, then antibiotics can be stopped. It's also been suggested that maybe the best use would be, because of the high cost of procalcitonin, procalcitonin can help you decide whether to give antibiotics or not. And CRP, because it is slightly non-specific, it may not be that useful in saying whether you need to give antibiotics or not. But CRP will, if a person has recovered, then CRP will come back to normal. So CRP could be used to help you decide when to stop antibiotics rather than using procalcitonin. So by using procalcitonin, you can decide whether you're dealing with bacterial infection or viral infection. And by using CRP, you can decide when to stop giving antibiotics in case you feel you are dealing with bacterial infection. I thank you for your attention. This is all that I wanted to say for the time being on respiratory tract infection.
Speakers for sharing their knowledge with respect to these two useful topics. Hope we have got now a better idea basically in aspect how the laboratory test works actually in the laboratory. Sir has shown a very good animated videos. With the help of that, we have got a better idea and a very clear picture how the laboratory tests on not only COVID, but even for other influenzas or even for the diagnosis of other complex pneumonias where multiple pathogens can be detected at the same time. How the technology works and how the system or how the PCR system works, actually we have got a very good idea. And let me thank to both the speakers for giving us this information and which has increased our means the scenario in both the respect in the laboratory diagnosis as well as in the clinical presentation. So I'm thankful for both the speakers. I'm thankful to the management of Meenakshi Labs for providing this opportunity for all our referral doctors for this knowledge sharing platform. I'm thankful for all the participants, our referral doctors, our marketing team who has done a lot of effort to promote this webinar, and all the participants who have attended this webinar, and hope it will be very useful for them. And finally, I'm thankful for our technical team who has help us to make this virtual platform very accessible for so that it is accessible to everybody. One more thing I would like to add, just regarding the Meenakshi Labs. Meenakshi Labs is a fastest growing laboratory chain, not only in South Tamil Nadu, but now we are planning to expand throughout Tamil Nadu, and we have now more than 10 branches which are functioning, which are not only collection center, but even we are doing this basic even inflammatory markers at around all our branches like CRP, Pro, calcitonin, and even both the speakers have already spoke about the importance of all these inflammatory markers, whereas D-dimers and other inflammatory markers for the diagnosis of COVID and even the post-COVID symptoms. We have facility available for all these centers regarding the swab collection facility. We have a collection booth at all these 10 branches where the samples will be collected for COVID and other inflammatory markers and other specialized tests. The samples will be get transported in the central laboratory for COVID, and here we have a short turnaround time where the COVID reports or PCR reports can be obtained within a period of 12 to 16 hours. So I would like to conclude with this, and I would like to again thank all the speakers and all the participants for making this webinar a good knowledge sharing platform. Thank you all.